Hello everyone. Today we are going to learn about file formats in bioinformatics. So first of all, let's start from the central dogma of life. We all know that DNA is converted into mRNA through a process called as transcription inside the nucleus, and then mRNA is converted into protein via a process called as translation, and that happens in cytoplasm. So when this data is collected from the cell, it becomes a DNA oriented data and a protein oriented data. And these DNA and protein data needs to be stored somewhere. So we store them in their string views. These are the string views of DNA. You can see DNA transcribing into RNA and then mRNA translating into protein. These alphabets, which are called as bases in case of DNA, and these alphabets which are colored here called as amino acids in case of protein, are stored in biological databases. So let's understand what is a database first of all. Database is a structured, organized set of data. And in computing terminology, a database refers to a software used to store and organize data. So you can think of it as a file cabinet where you store data in different sections called as tables. So whenever you will want to look at a particular data, all you have to do is write the keyword through which you can reach to that data. I have done a separate video on the unique identifiers, which are also called as accession numbers. Please check that out. So if now I want to access any DNA sequence or any protein sequence of my choice, and if I remember their accession numbers, I can directly write their accession numbers in the respective database and I can land up to my sequence. So now let's talk about the types of biological databases. So today we are only covering sequence database. I would like to remind you here again that there are various ways in which we categorize our biological databases in bioinformatics. I have done a separate video on that too. Please check that out. So now today we are only going to talk about sequence data which comes from either proteomics or from genomics, which means we will either have a protein sequence or a nucleotide sequence. So we have already covered nucleotide databases in earlier videos, and we know that these three databases, Genbank, EMBL, and DDBJ, they exchange the nucleotide sequences amongst them. And this is how a nucleotide sequence looks in a database. So yes, folks, we are going to talk about these type of formats in this video. And if talk about protein databases, you can visit some protein databases like UniProt, etc. And when you will reach there, this is how the sequence of a protein is going to look like. You can call it as a string view of both DNA as well as protein. Now let's talk about what is a file format. So file format is a standard way that information is encoded for storage in a computer file, right? And what is called as a sequence format now? So a sequence format, both for DNA and protein, defines the permitted layout and context of text in a file. And this includes text tokens that define fields used in a data bank. So these fields include the sequence itself, the sequence identifier name, and accession number, amongst other. So I am going to show you a few sequence file formats, and these are all the things which you are going to see in the formats. So let me take you to the place wherein I went to make this video. So NCBI we have already covered in another video, so please check that out. So we have come to NCBI again, and you can see this place called as database here. So you'll have to come here and you'll have to choose nucleotide from this window, and then I wrote insulin here, then I searched it, and then I clicked this sequence. So right now I wanted to have any gene sequence of insulin, and the organism I have chosen for today's explanation is Octodon decus. So when I clicked it, I retrieved its sequence. but its sequence came to me in a variety of formats. So let us discuss some of the formats before we see that. First is plain sequence format, which means that it is not in any sort of format. So you can call it as a non-format way of representing your sequence. Sometimes people call it as raw sequence, and we call it as plain sequence format. Another is GCG format. So like I have told you, these formats are sequence formats. So at the end, you are definitely going to see the sequences here. But before you reach to the sequence, you are going to find several identifiers which are required to be in that format, because we have different softwares in bioinformatics, and you end up taking your sequence from this database to another software, then to another software, and then you cannot keep on worrying about the conversion of the formats, right? So many databases, they provide you sequences in plenty of formats already. So the sequence file in GCG format contains exactly one sequence, which begins with annotation line, and at the start of the sequence is marked with a line ending with two dots. So GCG stands for Genetics Computer Group format, and these type of formats are usually worked out when you are working with a GCG package. So now, this is what I wanted you to see. This is the same sequence we were talking about. I chose octodon insulin gene sequence for this entry. So you can see that by default, it has opened in a format called as Genbank. And when I click this drop down menu, I see that NCBI itself is providing you the same sequence in different types of formats. So why do we need so many formats? The reason why we need so many formats is because there are many softwares present online, hosted by different types of resources and websites and web pages, which demand a sequence in a particular format for them to work on that sequence. So that is why we are now in need of softwares which can, convert our sequence in different sort of formats. And Genbank, at least, it is providing you these many formats, and you can convert it right here at this portal. So the first format I have chosen is Fasta. It is also called as FASTA, and the extensions are file.fa, file.fasta, and file.fsa. You will find this sequence and this format very easy to understand. The first line is called as the header, and the second line has the sequence. That's all. If you want to convert it into a raw, you just have to remove the first line, which is the header or the comment line, and then you are done. You will be left with the sequence. So this format is basically favored by many of these softwares because it is very small, concise, and you can store multiple data in this format, and it will not take much of the space also. Another format is called as multi-FASTA format, which is nothing but just the concatenation of all other many different FASTA sequences together. So this is how it looks. And the other is FASTQ format. Its file extensions are file.fastq, file.sanfastq, file.fq. And this is the example we have in here. So it has 1, 2, 3, and 4 lines in four different colors. So this format was developed by Sanger Institute in order to group together sequences and its quality scores. And the reason why they did that was because Sanger Institute is famous for genome sequencing. So they actually developed it to keep a tab on the information, whether the sequencing they did was of good quality or not. So this is what they wanted to know and wanted to pin down. So this is why they came up with this format. So Q here basically stands for quality. So fast Q is fast quality. So line 1, you can see here in orange, starts with the @ symbol, and it is a sequence identifier, and it is an optional description. So let's see what is the optional description here. So for this line particularly, let's see here. This is the unique instrument name. 208 is the run ID. This is flow cell ID. This is flow cell lane. So if you will see it, you will understand that this is the information regarding the wet lab experiment and the instrument they used for the sequencing of this genomic content. So the first line is totally comment. Second line, which is in green color, is the actual DNA sequence. Line 3, which says plus, is a comment line. If you want to add some comment to it, you can. And right now in our example, it has kept blank. So now let's talk about line number 4, which is in blue color. So this is a quality score line for each base pair, and this quality score is also called as Phred score. And this indicates how confident we can be that base was sequenced and identified correctly during genome sequencing project. So if you have a Phred quality score of 10, that means that the accuracy of having that particular base pair in our DNA sequence is 90%. And if it is 50, that means we are very sure that the genome sequencing machine has worked well, and the base pair we are looking at right now is the actual genuine base pair at that location. So if you want to know further, this is how it looks. So you can see here on the left hand side that below every DNA base pair, they have written these alphabets. So these alphabets have the meaning. For example, below A, they have written A. So if you can find out A here, so you will see A stands for 32 Phred score, right? Which is not very good. F here below A and T stands for, let me find it out for you. F is 37, which is also not very good. So likewise, J is 41, not very good again. So this is how, you get to know the quality of the sequence which is in front of you right now. But this is basically for them who are involved in genome sequencing projects. So now let's look at the Genbank format. So while I was explaining you how to dig out a sequence from the NCBI, so I told you that when we click it, by default we land up into Genbank format. So this is how a Genbank format looks. Starts with locus, have definition column, then an accession column, then the version column, then the keywords column, source, organism, reference, and like that. So I have discussed all these or the definitions of all these keywords in a separate video. Please check that out. In this video, I'm only explaining it to you how the format looks like. So the Genbank format is little descriptive, and you can see the actual sequence is coming at the end and under this keyword which is named origin. So you will see they have written 161, 121, 181. So you might be wondering what are these numbers. So the explanation is very simple. From this G to this G, these are actually 10 amino acids. The 10, 20, 30, 40, 50, 60. So in a line, they have 60 amino acids. So this T in the next line is actually the 61st amino acid. So just for the sake of, counting very easily how many base pairs we have, if one wants to do, this is how they can do. Otherwise, just be smart. You don't need to do that. You can read it here. But if for some purpose you are, you are dealing with something and you wish to know about a particular amino acid at a particular location, you want to count the location, this is wherein it can come handy. And this Genbank format is quite elaborate. It tells you which type of signaling peptides your sequence have, and what mature peptide will come from here, and what that mature peptide will make a protein eventually. So you can see it is giving you the the overall thing about the DNA contig you're looking at right now. This one is called as EMBL format, looks exactly like the Genbank format. It is also too much descriptive. It also tells you about the DNA you are looking at. It is also showing you the translation. Genbank also does it for you. And the sequence is at the end. And this is how you are going to understand it. Genbank is a database in NCBI. EMBL is also a nucleotide database. So these are two different databases from wherein I pulled these entries. So now the other format is called as cluster format, and the cluster format can be downloaded from the softwares which do multiple sequence alignment for you. So this is how multiple sequence alignment looks like. So now this multiple sequence alignment can be taken forward for creating logos or for creating the patterns or fuzzy expressions. So this is why the multiple sequence alignment formats are very important. So this one particularly is called as cluster format. So now let's talk about whether you have something which can help you in converting the format. For example, I have a format in Fasta and I want to convert it into Genbank. How do I do that? So we have a software called as Secret, and it is in the Emboss library. So Emboss is maintained by EBI and EMBL together, and they provide you multiple softwares to work with, which I'll be covering in my next videos. So for now, if anyone is interested, you can put your protein here, and then you can work with different type of formats. It can convert many formats to some other type of format. So earlier Emboss had another software called as redsec. So now redsec has retired, it doesn't work anymore. So Secret is the one which is working so far. So thanks for watching, guys. Please watch other videos also. I'll be posting more videos soon. Please write in the comment section if you want me to do some particular lecture for you. I'll get back to you with that. All right. Please share, subscribe, and like all the videos. Thank you.